My name is Caroline Ross and I'm from the LncLOOM lab at the Weizmann Institute. Today I'm going to talk about LncLOOM, which is a graph-based approach that we have developed to search for conserved elements in long non-coding RNAs. It is now well established that certain lincRNAs regulate key cellular processes. And although tens of thousands of lincRNAs have now been identified, only a very small fraction of these have been implicated in a specific cellular process. And it is not known how many of these lincRNAs are actually functional and how many are not. The major challenge is that lincRNAs evolve very rapidly, such that their sequences do not contain stretches that have significant similarity, and this makes it very difficult to use traditional comparative tools to predict their function or to classify different lincRNAs. So what is needed is a new approach that can detect highly conserved elements within these rapidly evolving sequences. Detailed studies of selected lincRNAs have indicated that their function may be attributed to short conserved regions that interact with proteins or other RNAs, and that these short conserved regions are disguised within this rapid evolution. So based on this idea, we have developed the LncLOOM framework. It's a graph-based approach that uses integer linear programming to search for combinations of short conserved motifs. The input to LncLOOM is a set of sequences which we then order as a set of layers based on increasing evolutionary distance, and this we use for the graph construction. LncLOOM then searches for motifs that appear in the same order, combinations of motifs that appear in the same order at different depths within this graph. We then compare these conserved elements to binding sites of microRNAs, and we use ECLIP data to determine if these elements may correspond to sites of RNA binding proteins. We hope to use LncLOOM on a broad scale to classify functional classes of lincRNAs and to use these combinations of short conserved motifs to detect lincRNA homologs in distant species. So LncLOOM uses a graph-based approach in which each sequence is modeled as a layer, which consists of a set of nodes where each node represents a chimer. These layers are then ordered according to evolutionary distance, and edges are then constructed that connect identical chimers between consecutive layers. This forms the basis of an integer linear programming problem in which edges can have a value of 1 or 0, 1 if they are allowed, 0 if they are not allowed, and we can then solve this subject to a set of constraints such that we want to maximize the sum of the edges. The constraints that we impose on the system are to achieve two objectives. Firstly, we want to select motifs that appear in the same order, therefore we do not allow edges to intersect. And we also want LncLOOM to favor long paths of more conserved motifs over motifs that maybe are more repetitive but are not conserved in more distantly related species. So we set up our constraints such that any path that is selected must be fully connected, and we can then solve for our combination of short conserved motifs. This is an example of what we would expect to get from this input. The significance of our motifs are then empirically determined, and we do this by running multiple LncLOOM iterations on random datasets. Firstly, we want to determine the probability of finding the exact motif at the same depth, or to the probability of finding a similar motif of the same length at the same depth. And we want to do this in random datasets that firstly have the same percentage identity, which we calculate by shuffling multiple sequence alignments of our input sequences, as well as looking for these motifs, what's the probability of them occurring in random sequences with the same dinucleotide composition. To show the To demonstrate the power of LncLOOM, we performed an analysis of Serrano and chaser lincRNAs. The Serrano lincRNA is broadly expressed, but the sequences vary significantly. So, for example, if you run a BLAST between the human and zebrafish Serrano sequences, BLAST will only identify a very short region of 67 nucleotides. Further, if you look at the 100-way whole genome alignment, there is no alignment is detected between the loci of mammals and fish Serrano species. So we performed our LncLOOM analysis on Serrano sequences from 18 species, including mammalian and fish, as well as elephant shark. And what we found is that LncLOOM does detect highly conserved elements both within this region that is alignable by BLAST, but also outside it. And you can see that these conserved elements do correspond to microRNA binding sites or an RB-Fox site and an open reading frame. Our chaser analysis was performed on sequences from 16 vertebrate species. Again, BLAST-N does not report any significant alignments between human and several vertebrate species, and no homology is detected between human and zebrafish. And we show that LncLOOM really was capable of identifying conserved elements. And our next step now, which we are busy with, is to now perform experimental validation and to show that these conserved elements are actually play a role in modulating the function of chaser. We also want to perform large-scale analysis of other lincRNAs, 3' UTRs, and enhancers, and we want to extend our LncLOOM framework to look for conserved regions, conserved elements in monophyletic groups. I would just like to acknowledge Igor Litsky, he's my PI, and all the members of his lab and everyone that has been involved in this project. Thank you.